So let's see. Welcome, everybody. This is our third panel for the RCN-ECS Spring Reading Group for 2023. And today we're going to be talking about applications of gene editing for conservation. And I just wanted to introduce our three panelists for this session, and thank you all for coming, especially Jackson, who's on a very different time zone, so it's 11 p.m. for him. So I'm going to just give a quick introduction for each of you, and then if you want to just give a description of your research and how it touches on the topic of gene editing for conservation, then we can kind of start, I've got some questions for the panelists so we can kind of have a discussion amongst ourselves. And we can also have questions from the audience, especially if more people show up. But I'll start with you, Jackson. So our first panelist is Jackson Champer. He's a faculty member at the School of Life Sciences at Peking University and at the Peking Tsinghua Center for Life Sciences, and also a member of the Center for Bioinformatics there. So welcome, Jackson.
Thanks. So my research is all about gene drive, which is a way for engineered alleles to bias their inheritance and potentially be passed on to all offspring instead of just half, as you would get from a normal allele. This means that if you release a few gene drive individuals, that gene drive allele can then spread throughout the population. It can be used to modify or suppress the population. So my research is just everything about gene drive, designing it, testing new things in flies, eventually getting things working in mosquitoes, as well as computational modeling of gene drive to find out what would actually happen if you released one into the environment.
Okay, cool. All right, thanks, Jackson. And then, so next we have Phillip Cleves. So Phillip is
It's Cleves. My last name's Cleves.
Okay, sorry. Cleves. All right, gotcha. Thank you. Sorry about that. So yeah, Phillip is a principal investigator at the Carnegie Institution and in the Department of Embryology and at Johns Hopkins University. So Phillip, you can take it away.
We are working on corals. Coral reefs are biodiversity hotspots of immense importance. However, they're threatened by climate change, where when water temperatures get 1 or 2 degrees higher than what a coral typically sees, they'll bleach and lose their algal symbionts. And if that water temperature stays high, they'll die. Over the past several decades, we've lost a lot of reefs, including 30% of the Great Barrier Reef in 2016, in a course of a few weeks, really. Despite this, we know very little about the genetic and molecular basis for why this happens. Indeed, there's a lot of arguments still about why corals bleach and what types of genes are involved. And so in our research, we've built the ability to genetically modify corals for the first time with CRISPR. And our research goal is to continue to build those tools to try to understand why corals bleach and if there are any genetic determinants for why some corals bleach and other corals don't, so that we can use that information to guide conservation efforts.
Okay, cool. All right, thank you, Phillip. And then last but not least, Mike Samuel is a professor emeritus at University of Wisconsin-Madison, was in the Department of Forest and Wildlife Ecology there, and actually kind of met Mike there when I was doing my PhD, and I was looking through some of the sorry about that. And I was looking through some of the literature, and I was really interested to see that he, in some of his work on disease ecology, had published a paper on the potential for using gene editing to combat avian malaria in Hawaiian forest birds. So, Mike, if you want to talk about that a little bit and your generally that research project, that would be really cool.
Sure. I'm basically a disease ecologist. I'm not a geneticist. So my sort of interest is in applying genetics where it's feasible or where it's possible into some of the wildlife disease systems. So I've been involved or was involved with the Hawaii system for probably 15 to 20 years. Starting in the mid 90s, we got a very large grant from Biocomplexity to try to understand the malaria forest bird issue there. Those birds evolved without malaria and without mosquitoes, and so they're very susceptible to that disease, which was introduced by people coming over in ships and dumping water and things like that at casts and introducing mosquitoes and later malaria. So the mortality rates in some of the species are as high as 90% once they get infected. So as basically the context is within climate change. Currently, low elevations are highly affected by malaria and mosquitoes. And there's only one native bird that still remains there, which has evolved some tolerance. At mid elevations, there's quite a bit of malaria transmission, and it's pretty low and seasonal at higher elevations. But as climate changes and warms, the mosquito malaria cycle will move up the mountains, and extinctions are predicted for many of these species, which are not found anywhere else in the world. So we've been involved in trying to understand that system for quite some time. So we did sort of a hypothetical simulation of how or whether when those birds, particularly Eevee, which is a very iconic bird in Hawaii, it might be possible to do CRISPR editing to give it resistance to malaria, and how that might then play out in terms of introductions and success of the resistant birds becoming more dominant in the population.
Okay, cool. All right, thanks, Mike. Okay, so yeah, I've got a couple of questions for you guys. And let's see. I think I'm just gonna start off with something that I think was that's kind of generally interesting to me in a lot of this work, and in the paper that we read for this week on gene editing, which was I don't think actually any of you guys were on the paper, but it was also on using gene drives to potentially eliminate invasive mice on islands. But I feel like there's a fairly strong theme in this gene editing work on ecological interactions, either kind of targeting maybe disease vectors or hosts to try and slow or halt the spread of disease, or targeting invasive species to try and eliminate them as a threat to native species. Also, the corals kind of naturally lend themselves to this kind of ecological interaction. There's a host animal, and then there are the symbiotic dinoflagellates. So that means, though, that we have a couple of different targets for gene editing. The host, or the native species, or the vector, or the invasive species, or the symbiote. What do you guys think are the kind of pros and cons of targeting different members or different kind of components of those interactions with gene drive? Is it better, or more effective to target hosts or vectors or things like that? And how does that kind of how do the different study systems that you're working on, how do those kind of affect, which one is more efficient to target?
Well, I think the answer to that is going to very much depend on your scenario. From my perspective, a gene drive, for example, could be used to deal with the situation of avian malaria in multiple ways. The simplest gene drive is modification drive, which could spread a cargo to prevent these mosquitoes from transmitting the malaria, either to birds or to humans. We're actually studying both Anopheles mosquitoes, which transmit malaria to humans, and we're also trying to make a better gene drive in Culex mosquitoes, which are those nasty guys in Hawaii. For modification, you need a specialized cargo that will do what you want. In this case, it's something that could go after the malaria in the mosquito. In other situations, a suppression gene drive might be more suitable, especially for something like an invasive species that's not supposed to be there in the first place. On the other hand, if it's an important species that's causing a problem, then you might need to find a modification system. And there might also be nothing to suppress, such as the coral reefs. It's pretty much them against rising temperatures. There's nothing really to suppress there that's threatening them, at least not that I'm aware of. But you would need some way to modify them, and a gene drive could potentially allow you to spread high temperature resistance genes to a population more quickly than just genetically modifying them and then releasing a few of them. You can keep more genetic diversity in the population, for example, with the homing type gene drive. So it's extremely situation dependent, both the situation and the tools you potentially have available.
We're not doing gene drive, and we're not even considering doing any genetic modification in corals to release them into the wild. Our research interests are to try to use genetics to try to figure out what the basic biology of these animals, not to do genetic interventions. I think we're really at a rudimentary understanding of these organisms. They have huge population sizes with large amounts of genetic diversity that exists within these populations that are being selected by natural selection from these warming water temperatures. And so our goal is to do exactly what has been done in biomedicine research for the past 50 years, and try to use the ability to manipulate the genomes to try to figure out how the organism works. Because we think that if we understand the more basic what's happening, what makes corals more resistant or less resistant, then we can come up with at least insights that might guide novel conservation efforts. But the idea of these kind of modifications being put out onto reefs, I think, is not something that we're interested in really even entertaining.
So I would say in the Hawaii system, there's some complexities to overcome. I think certainly treatment of mosquitoes is probably the easiest, not the easiest, but probably the most, let's say, socially acceptable strategy. There's been a lot of issues about using GMO organisms in Hawaii already. And there's a lot of, I guess, the spiritual connection with the native Hawaiian people and most of the wildlife and species and plants and species there. So that's a social stumbling block. And probably mosquitoes are a lot easier to work on and a lot easier to modify than it would be to worry about what's affecting immunity in a wild bird species. Since at this point we haven't even identified what genes might be involved in immunity. So I would tend to think that mosquito strategies like gene drives, what they're doing now currently is they're looking at Wolbachia bacteria as a way to try to control mosquitoes. The complex challenges it's a landscape level issue, and there are a lot of mosquitoes out there that any of these strategies have to work at a very large scale, and are likely going to take some time.
I will say one thing along those lines with the mosquito question. For these discussions, it's also important to realize what the baseline is. So, in medical research, when you design a therapeutic, it has to be better than the current standard of care. And so I think that's a concept that I think would be really important for are thinking about gene driving mosquitoes. What is the standard of care right now to manage mosquitoes? What are the costs of doing that? And what are the pros of doing that? And then does any kind of genetic manipulation alleviate those costs or introduce additional problems? And that kind of cost benefit analysis with the current standard, not the ideal, I think is an important place to start.
I certainly agree with that. In fact, there can be even some complex considerations for other methods of genetic control. For example, a popular one is to release a lot of sterile males. Then the females mate with the sterile males, don't have offspring, so the population is suppressed. There's no gene drive because the sterility alleles don't themselves spread, or at least not significantly. But it's still a potential method of genetic control. It's often worked pretty well in the past, and newer methods are a little more powerful than the old ones. But in a recent test, I think one that was actually based on Wolbachia methods, the ones that were released were sensitive to insecticides, which was the current standard of care for the mosquitoes in that time. So needless to say, the efficiency of that intervention was quite a lot lower than predicted until they started using mosquitoes in the native background that had a similar level of insecticide resistance to the wild ones. So something like this shows that you could have complementary treatments if that would still be cost effective, but only if you make sure the treatments aren't interfering with each other.
Okay, great. Cool. I think another a very kind of basic question, I think, and I think Phillip is definitely more on working on this application of it. Would just be how these methods can help us understand the basic biology of the organisms that we're interested in. And I would say kind of a complementary question to that is like, how much or what do we need to know about the organisms? How much more do we need to know to be able to apply these methods to non-model organisms? Because I think that's one stumbling block that has come up for things like, Mike's potential plan to kind of alter resistance dynamics in the host itself. So just kind of what do we need to know, and how does this help us potentially know that about our species of conservation concern?
Yeah, I can. So I mean, there's a rich history of using genetics in a handful of model organisms. Indeed, most of making random mutations in organisms that had biology that were easily tractable in the lab, and then studying those random mutations, and that's was before genetics. And then reverse genetics emerged with the ability to manipulate the genomes directly of organisms, again only a handful that were tractable. And I would say much of what we know about what genes do in any organism come from these types of either forward or reverse genetic stuff. But we're in a emergence of several types of technologies, TALENs and then CRISPRs, that allow us to make modify the genomes of organisms across the tree of life. Butterflies, single cell protists, corals, mosquitoes, right? And so it really has unleashed the ability for us to genetically study like the diversity of life, which offers an incredible opportunity. It's also daunting because there's so many interesting questions in biology. Where do you start? You know, is it going to be corals, or is it going to be trees, or is it going to, you know, and so I think that this is like kind of something that the next generation or current generation of scientists grapple with. How are we going to focus? Do we need to focus our efforts to study the genes that matter in important systems? You know, taking the lessons, we are taking the lessons from this, you know, era when we could only study a few model systems at the genetic level in any reasonable detail, and now applying it to questions that we think are the most important for conservation. And corals, for us, are a very charismatic and threatened species that are facing an extinction crisis. And so we think that applying the exact same this intellectual framework of like, let's break a gene, see what happens. Let's study like why corals bleach, why some don't. We think that just like it was important to understand the genetic basis of human disease, if we had a better understanding of these ecological problems at the genetic level, that would inspire new conservation strategies. Now, I would say that this is also still very basic science, right? And so we don't know, as scientists, we don't know what the future application is going to be for any individual discovery. I mean, CRISPR is a great example of that. The green fluorescent proteins, studying those from jellyfish, another great example of how we don't know where our discoveries will lead. But that when we have a better understanding of organisms that are important, that we are insight to be able to conserve them. And so that's the system, and that's kind of where we're focusing now. Where the second part of the question is, how do you make the transition from model system to non-model system and bring technologies across? And so my perspective on this is one where it's involves interdisciplinary science. So my background was in cell and developmental biology in fish. We were studying, I was in my PhD, and I moved into corals as my postdoc, and I collaborated with people including Lena Bay at the Australian Institute for Marine Science, who was an expert at coral reproduction and coral ecology and coral genetics, really. And so by fusing this collaboration, I brought the ability to do the molecular biology and the CRISPR with her ability to know, you know, the organisms out in the field, and by that interdisciplinary to genetically modify these organisms. And so I think a similar strategy of merging organismal biology with molecular biology through collaborations is probably a fair approach to do it.
To me, there's a lot of steps to go through, things like endangered species, or other wildlife species that don't lend themselves to a lot of laboratory study. And in many cases, I'm not sure we even understand what the specific genes are that need to be, that even are even identified yet. I mean, there's efforts to try to somehow engineer plague-resistant other types of things like that, which you know, are going to involve a very long time and a very complex sort of approach to tradition, where you can't really, because years and reproduction is an issue, and are we affecting fitness when we do any of these manipulations, and lots of complexities that are likely to be the case in the real world for those organisms.
In general, I pretty much agree. Even from a gene drive perspective, having these be essential. You know, I would never even try to make a gene drive in an organism that already didn't have a lot of things known. You know, some are things that, you know we would want, you know, the function of genes, expression patterns, this sort of thing. Bugs are things that you wouldn't necessarily be able to, predict just from some random starting generic situation in an organism you knew nothing about. In mosquitoes, for example, you could just put in an anti-malarial gene, but if you knew enough about an extremely targeted attack on a particular mosquito gene that malaria or dengue or something like that needs, air modeling has shown that something of that sort would actually make for a far more efficient and longer lasting drive approach. So, basic science is necessary not only for making gene drives in the first place, but for making gene drives that have the best chance of actually accomplishing something positive.
Great. Okay. Yeah, that kind of leads into, I think, one of another question that I had, which is, since some of this stuff is still science fiction, I think there's a lot of modeling involved in these early stages where, we can't actually release things into the wild or even, find the genes that we need to edit. But how important is modeling, and how can we use it to best kind of target these interventions and make them, more efficient and more likely to succeed?
Well, from a gene drive perspective, modeling is really important for two things. The first is finding out if the gene drive is good enough to get the job done, whatever that job may be. And the second is to make sure the gene drive won't do anything we don't want it to. Many types of gene drives, such as the most well-known homing drive and X shredders, which are in the Journal Club paper, are unconfined gene drives. That means with even a very small level of migration, they will spread potentially uncontrollably in the population. There are many options for a confined gene drive, but you don't just choose a confined gene drive and then all of a sudden you're confined. No, there's many different levels of confinement, and choosing confinement is going to be a balance between confinement and strength of the gene drive. If you want more confinement, you're going to have to make a larger effort. So being able to predict the level of confinement needed, the type of gene drive that will work best, is going to be very important for knowing if there's a gene drive solution, and if so, exactly how to deploy it.
Sort of fail-safe mechanisms from gene drives that maybe don't give the expected results and need to be, terminated, basically. Is that something you guys think about and work on as part of the gene drive system?
To a certain extent. So there's lots of things people have thought of, like, kill switches based on some type of pesticide intervention or pesticide-like intervention, or the most promising is probably an overriding gene drive, kind of a gene drive that targets the undesired gene drive and can thus eliminate it. But all of these things have complexities, especially for the uncontrolled gene drives, which could potentially spread through the entire population before these reverse drives could catch up to it. So, we would prefer to kind of keep the initial tests of a gene drive confined in the first place, rather than kind of be scrambling to contain something that can theoretically spread everywhere in the world that the population exists. So we do think about these things quite a lot, and what the advantages and disadvantages of the different methods are. There's already, several publications from different labs looking at these sorts of things. So there are options, but not necessarily always good options for this.
Yeah, my view on organisms out into the wild to contribute to it.
So I guess one of the things I'd say is that, the modeling stuff we did was hypothetical. I think, and lacks a lot of probably important information. There's, we just had to make assumptions about how things might work in terms of fitness and reproduction and, breeding with other genetic types. I think there were still some things that we learned by doing it. I think I was surprised to see in our simulations that a component of whether we'd have success or not was how much genetic stress was put on. In other words, how much malaria transmission was going on. Predictions were that we would do better when there was more malaria being transmitted, because it would favor the resistant types. And that was an important part of what might lead us to success. So I think there can be, depending on your understanding of the system and how well you can model it, I think there can be some surprises that are, you wouldn't predict if you were just sitting down and thinking about it. Or certainly people who are trying to implement those systems, managers, let's say, in the case of the Hawaii system, probably would not think about in terms of how they might think about implementing the system.
And that's certainly true. Air modeling has also discovered some quite unexpected phenomenon that have kind of changed what we viewed as the original requirements of what would work.
Could you elaborate a little bit on that? What was unexpected about that?
Sure. So this was mentioned a little bit in the journal club paper. We call it chasing, and it applies to a population suppression drive. So this is when the gene drive will locally eradicate a population, let's say in the middle of a big patch. But then, instead of going on to eradicate the rest of the population, some wild type individuals manage to sneak into the empty area that the gene drive has cleared. In this empty area, these are insects we're talking about. So they're able to have tons and tons of offspring because there's so much less competition. This is especially true for mosquitoes, where the larvae intensively compete for resources in pools. So this means that now the wild type population is blooming again in this empty area. The gene drive is still present, so it can kind of swoop back in and start suppressing the population again. The wild type population can always stay a step ahead of the gene drive, never quite catching up. So we found that in a panmictic population, a simple environment, the gene drive only needs a limited amount of power to suppress the population. But in this spatial environment, that was never enough power. It needed to either be much more efficient, or the ecological parameters needed to be just right to prevent this chasing phenomenon from dominating the situation. And this has some interesting interactions, because even if the population goes down a lot during chasing, it requires constant gene drive activity. And for all the gene drives so far, whenever there's gene drive activity, there's the chance that we might get functional resistance alleles, which is basically a version of the wild type allele with a slightly changed sequence that prevents the gene drive from working properly. And if we get functional resistance alleles, it's game over for the suppression drive. So even if chasing was successful in reducing the population down to an acceptable level, that might only be a temporary situation because of this resistance, even though in a simple panmictic population, there wouldn't have been time for resistance to form, and the gene drive would have been completely successful.
I think your spatial conclusions are really interesting. Is the solution to that to, rather than do spot treatment, to do sort of more spatially global treatment or introduction of gene drive mosquitoes? Solution that you found?
Well, our investigations in this are still pretty early. We actually did try a widespread release, but it's such a stochastic process that you can still often form some small pockets here and there of wild type and still start the chasing. It did kind of reduce a little bit the initiation of chasing, but it didn't completely change the situation to stop it from being a problem. However, we have recently found that in more ecological rich environments where there's competing species, that might be able to prevent this chasing phenomenon if there's enough of an ecological niche overlap that when you get rid of one species, another can at least partially take its place, which will inhibit then the original species coming back into that area, at least long enough for the gene drive to catch up and eliminate the population. This has also presented other issues like stochastic loss of the gene drive, which increases in these conditions. So rather than a widespread release, in these conditions, it might be best to every now and then do a small follow-up release just to ensure that your gene drive isn't stochastically lost from the population. So the gene drive can kind of work together with the competing species to eliminate your target population. We've recently been considering this in the context of invasive fire ants, which are almost always competing with native ants, usually outcompeting them, but still competing. It's also the case in mosquitoes, though. There's maybe about 10 or so mosquitoes that go after us, but thousands of other mosquitoes in the similar environments that, as far as we know, at least, aren't causing too much harm, since they're adapted to the ecosystem and not specialized on humans, which have rather grown in population compared to when most of these organisms evolved.
Yeah, it's interesting that from an ecological perspective, I mean, you could eliminate one species which favors another, which maybe also have its own set of deleterious effects that are unexpected.
Yes, I'm a bit worried that a suppression strategy in malaria mosquitoes might just open the way for another species of mosquitoes that transmits almost as much malaria, but that is normally outcompeted by the target species and thus usually beneath the notice of scientists.
Or may transmit another disease like West Nile virus or something like that as well. So, yeah.
Yes, I think like replacing Anopheles with Aedes, for example, probably wouldn't be too high on people's to-do list. So because of this, I'm still very interested in mosquito suppression, but it always makes me think, oh, maybe a modification strategy might be the simpler thing here, so we don't have to worry about completely changing the ecology of the situation.
Not to be completely focused on the Hawaii situation, but do you have thoughts on the Wolbachia strategy they're using in terms of those kinds of questions?
So Wolbachia is pretty good because when mosquitoes are infected with Wolbachia, they tend to be much less able to transmit diseases. It's also something that can actually spread like a gene drive. So these Wolbachia are transmitted by mothers to all of their offspring. And the males also have kind of a special mechanism. So just like with the gene drive, you can release Wolbachia into the population and then it can continue spreading on its own. I think if we could get these into culex and they do prevent malaria, it could be an excellent strategy. However, there's also another way that Wolbachia are used. Males infected with Wolbachia, whether they're mosquitoes or many, many other species of insects, actually can't have offspring with uninfected females. So it's kind of like a sterile insect technique strategy. Release tons of males with Wolbachia, and then the females will mate with them and not have offspring. This could be an effective way to suppress populations, but it does require very high release sizes and widespread release sizes. It might be more suitable for limited urban areas that are seeing lots of transmission rather than widespread areas. But Hawaii might be small enough and well-resourced enough to still pull it off. I haven't seen any specific modeling for what the large-scale requirements of that would be, though. So I'm a little hesitant to make such economics-based statements.
Wouldn't it have the same chase kind of issues that you talked about for gene drive?
So this type of release strategy, Wolbachia, as well as other stereo insect techniques like RIDL, Riddle, probably wouldn't suffer from this, because chasing in gene drive comes from, a single release and then natural evolution of the population. For these strategies, you're doing a widespread release everywhere all the time. So you might sometimes get some temporary stochastic survival of wild type, but presumably it wouldn't take too long for more released ones to come there in the next few weeks. So I think chasing is less of an issue for this type of strategy. The issue with this strategy is that it requires, thousands and thousands of times greater release size than a gene drive based strategy. If you release gene drive everywhere at these levels, chasing probably wouldn't be a problem either. For the same reasons, you're always overwhelming it in every spot rather than just having one spot of gene drives that's moving around.
And well, sorry, this is probably not where you wanted to go, Brendan, but it seems to me if those sort of sterile mosquito strategies also have to be applied for years and years and years over large scales in Hawaii, for sure, for example, and probably other places as well.
A very famous use of that, going after, what were they called? I think the New World screwworm, which is a major livestock pest, started, I think, in the 70s, roughly. And it started in North America and over the course of many, many decades worked its way all the way down to Panama, rolling back the invasion of this species, but then couldn't get any further because the species was too well entrenched in South America. So it shows that this kind of strategy, if you're willing to wait and be thorough, can be extremely successful, but that it also has its limitations.
Mike, I think this is a super interesting discussion. This is kind of where I hoped partly this panel would go, because I think just finding the overlap between these different approaches, and I think you're both coming from very different very different kind of starting points. Just hearing you guys talk about all this, all the intricacies and what would work and what wouldn't, I think is really interesting. Yeah, this is great. I was wondering, just to kind of switch gears a little bit, just thinking about how these technologies are so new and are potentially kind of have ethical implications that people haven't completely sorted through yet. Whether any of you guys have encountered resistance, or I mean either resistance or kind of enthusiasm from management partners or funders or governments for either using these technologies in the wild, or using them maybe like Phillip is using them to better understand the basic biology of these species. What has your reaction been? What are the kind of considerations that you have to take into account when you're pitching these ideas to funding organizations, and have you been surprised by any of those reactions?
I can start. I mean, I think that the use of genetic engineering in corals to understand how they work, I think, is attracting a lot of attention for the first principles aspect. I think there is a lot of confusion in the field about why corals bleach. There's several active and contrasting models for why that happens, and they're largely long-standing hypotheses that are really unresolved. And so I think there's a lot of interest and attraction to the ability to use genetics to actually ask and understand the basic principles in the first instance. And then, but there is a little bit of contrast, right? Because basic research takes a long time, and in this instance of corals, we have not very much time if the models are right to act and to help conserve these ecosystems. And so I think there is a little bit of I think that both things need to happen at the same time. We need to come up with effective ways to conserve and restore these ecosystems, but also simultaneously doing basic research so that we know that the things that we're doing are effective, and to inspire new strategies. And so depending on what side of that work you're on in corals, you may weigh the relative importance of those two things differently, considering the urgency of the matter. But I do think that the truth is, and probably the best strategy is in the middle. Do simultaneously push both basic research and practical conservation strategies, and let the two groups, hopefully they're not actually two groups, but let them make sure there's good communication between both efforts. Because it seems to me that we need, in the case for corals, we need a deeper understanding of just how corals work to be able to predict what's going to happen in the future, and then being able to predict what happens in the future can help focus those conservation efforts. And so I think that is some tension that I don't think needs to actually be there, because it's all towards the same effort.
So that makes perfect sense to me. And who is providing resources that would facilitate that kind of collaboration for the corals work in particular? It seems like we're sort of siloed in terms of how we provide both basic and applied research funds and get at these problems, and they don't tend to be in sync very frequently.
Yeah, and I think that there is a need for that type of intersectional funding. And I will say that there's, certain groups are more likely to fund one type of research versus the other type of research. And whether or not it would be more effective if there was kind of a intersection between those approaches, and that there are funding agencies or funding opportunities at that intersection, whether or not that would be more effective than Sutton. But I do think that in the coral space, it's well known that both our understanding of these organisms is rather limited, and it's also well known that we need to do something. We feel a strong urge to need to do something to help conserve them, given what's happening out in the field. And so I think that both those things are well understood by the community.
I've noticed that funding is often a very stochastic process, but I think the animal community is a little bit better about supporting basic research than perhaps the human health community, where I think complaints are very widespread about not having adequate funding for basic research, always trying to jump straight into an application. That's definitely unfortunate because of the need for widespread basic research, and that's what often leads to the highest impact future applications, just not necessarily right
or predictable.
However, even in GeneTribe, which is often always just one step away, I find that a lot of the time reviewers of grants don't really fully understand the modeling aspects of the grant, or even the need to try out some new gene drives and model organisms before moving straight into mosquitoes. So you're always going to run into a few potholes here and there. But at least there is a general agreement among many people that we need to fund the basic stuff. It's fundamental.
Yeah,
The other part of
I
your
have a little-
question, sorry.
Yeah, I was gonna say I have a little different perspective on how funding works. You know, I view funding as kind of a pyramid, and at the bottom is human health or human diseases. Now, this is my perspective as a disease ecologist. You know, the next level up, sort of that intermediate level, is veterinary sciences, basically domestic animals and livestock and those kind of things. And it is wildlife funding problems. And I think sort of some of the One Health perspective has had a little bit of change in that, but sort of my take on a lot of the One Health perspective is that, yeah, it's great as long as it has a human impact of some kind. And if it doesn't have a human connection, then, you know, it's more of a conservation issue, and you're back to sort of that sort of small part of the pyramid, as far as I can tell.
I do think that the winds may be changing on that. I mean, I think that of course, there's going to be a lot of money that goes into human health research, but I do think that there is an understanding that's emerging as we see our climate change, that environmental health is human health. And I think that there are NIH initiatives that directly link that try to say how our environmental pollution is affecting human health. And I think that that's one step toward dismantling the pyramid, as you said. I think that's a nice way to think about it. And I do think as we continue to see pests and ecosystems degrade and water issues, I do think that we'll start to see a change in that view.
Well, it'll be interesting to see if humans are willing to change their footprint in terms of its coming down on the most of the ecosystem enough that it actually makes a difference.
System who is try to check the impact of global warming. And also it's important that in corals to think about the fact that they are living animals that are complex, that have the capacity for us all together, that we come from a long line and reproducers that have faced hardship in the past. And so I think that you know, positioning these ecosystems in a way where they can undergo naturality more successful than anything that we can try to engineer in the lab.
Yeah, if I could follow up on that just a little bit. So another system that I've been heavily involved in for the past 20 years is the deer prion disease called chronic wasting disease. And we have very strong evidence that there's a genetic selection that's actually, and that the less susceptible genotypes are being favored at a pretty rapid rate. But when we did simulations of what the likely they're not exactly what the results of the simulations are not exactly what you think. While you develop more resistant or less susceptible genotypes is probably the best way to say it. It means that prevalence of the disease actually increases, because it's not that those animals don't get infected. It takes them longer to get infected, but they also live longer once they become infected. So there's more transmission that actually can occur. Just whether you've seen and potential good changes in terms of some of the coral issues as well.
I can answer that. I mean, that's a huge avenue for animals will adapt. There's pretty good evidence that corals will change their symbiont type based on the type of environment that they're in. I would still say there's a lot to be done about trying to model these in these large ecosystems, trying to incorporate the genotypes of the symbiont and the host, and try to come up with any predictive framework, especially considering, some of these ecosystems have hundreds of hard coral species that all have different demographics. And so I would, it's a really interesting, as a coral biologist, is one, as an ecosystem steward, of watching these organisms adapt to very strong selection pressures that are in the field, and how far ahead can we get in that prediction is kind of the golden question.
Great. Okay, thank you everyone. It's yeah, we're a little over an hour now. So I don't know if there are any other questions or like concluding comments that anyone wants to make. But
I had a sort of discussion we were having. So if you can identify, let's say, species that have adapted, would the public favor a strategy that's sort of capturing those genotypes and releasing them, or versus a gene edited approach? What do you, I mean, how do you use this, sort of, what's your thoughts about what might be socially most acceptable?
I think there's a lot of efforts currently being undergone in trying and just replanting corals that are naturally out there. I mean, huge efforts in the Florida Keys are trying to put out corals that have undergone massive population bottlenecks through disease and other stressors, bleaching. And so those efforts are already underway. I mean, in Australia, they're ramping up huge programs to try to outplant massive numbers of corals to as stock. And so, to me, particularly, the question is with those efforts is whether or not those corals that you outplant are going to survive the next heat wave or the next disease outbreak. And I think that that's a really interesting question that we need to solve. If effort's going to be eradicated in the next stress event. So it seems to me, if you could predict which ones may have the highest probability of survival, that those might be the ones that you would want to focus these already existing replanting efforts on. Would that lead to genetic bottleneck in the systems that would decrease like the ability for these organisms to adapt to future scenarios? Maybe. Is that any different than what's already happening in the field with, especially in the Keys, when you have, only a few animals existing in some regions? I think that that's a good question, and all these things need to be weighed when you're doing it. But there's already large replanting efforts that are happening to try to restore some of these ecosystems.
I wonder if you could do some kind of experimental laboratory evolution to try to kind of guide more variants in the lab. If you're able to get a faster generation time than out in the wild, you could then let evolution take its natural course, get something good to release, and maybe also get some high strength alleles after sequencing these things to see what the evolution was.
It's certainly something that's already being done right now for the algal symbionts, because those can grow quickly in culture in large population sizes. So there's groups in Australia that are trying to generate artificially selected algal symbionts, and we know the genotype of the symbionts also contribute to the bleaching tolerance of the animal. And so that's certainly something that's being worked on now. Doing that on the animal side in the lab is a little bit trickier because the generation time is about 3 years or 4 years. And so those types of experimental evolution experiments might be more appropriate just watching what's happening out in the field, where you have these huge reproductive events with large population sizes.
But I don't know where we are in terms of being done, but I have to say thank you. I learned a lot from you guys. It was good, good discussion, and I had
Yeah, me as well. You guys have very
things I
interesting
hadn't
thoughts.
thought about, so very helpful.
That was interesting. Thanks.
Yeah, great. Thank you everyone for joining, and yeah, have a great rest of your day, and this is a great discussion. So talk to you later. Bye everyone.
Good to see you, Brendan.